adaptive defenses. This is the part of the immune system that can learn about any foreign substance it comes into contact with, which we call antigens, and develop the ability to protect the body from that specific antigen anytime in the future. But how can your immune system have such an incredible memory? And what does this even mean? To understand this, we have to learn about antibodies. These are large Y-shaped proteins that are produced by lymphocytes, and they circulate in the blood and lymph, looking for pathogens, which they are able to mark such that phagocytes can recognize them for destruction. So what are these antigens? The word antigen is derived from the phrase antibody generating. So the word refers to any foreign substance that will be recognized as being not of the self, and will thus provoke an adaptive immune response. These can be proteins, polysaccharides, lipids, any large molecule that doesn't belong, as well as many pathogens, as these will bear foreign surface proteins that can also be recognized. The part of the foreign substance that interacts with the immune system is called the antigenic determinant. An antibody or lymphocyte will bind in a way that resembles enzyme-substrate interactions, and different lymphocytes will recognize different determinants. These include B lymphocytes or T lymphocytes, depending on what type of immunity they oversee. And there are also antigen-presenting cells. Lymphocytes of either variety originate inside red bone marrow from hematopoietic stem cells. They then become immunocompetent, meaning they gain the ability to recognize a particular antigen, and once committed to a particular antigen, thousands of surface receptors are produced that are devoted to that recognition. On B cells, these receptors are actually membrane-bound antibodies. These lymphocytes must recognize certain proteins, but also learn self-tolerance, meaning they must not attack the body itself. Cells that fail to do this are forced to undergo apoptosis, and in fact only about 2% of T cells will make it, but the ones that do will rapidly divide to make many copies of itself, all with the same antigen recognition. Now let's talk about two types of adaptive immune response. There is the humoral immune response and the cellular immune response. The humoral immune response occurs when a new B cell encounters its antigen, which causes endocytosis, followed by proliferation and differentiation into plasma cells. These will then mass produce the antibody that recognized the antigen, and these will circulate in the blood and lymph, looking for that same thing again. This is referred to as the primary immune response, and it takes a few days to make all those antibodies, which is one drawback to this defense strategy since it protects against future invasions, but it can't act so quickly upon the initial invasion. However, if that antigen does come back, the secondary immune response begins, and this will be swift and effective, with plenty of antibodies to tag the antigen for destruction. This kind of humoral immunity can be attained naturally through infection or artificially with vaccines, which allow for the primary immune response to occur with an inactive form of a pathogen so that if the real thing ever comes by, the immune system is already ready for it. In either case, we are describing active humoral immunity. Passive humoral immunity is different because the body doesn't go through the work of recognizing an antigen and generating antibodies. Instead, these antibodies can be introduced directly into the body, either through a mother's milk or through injection of gamma globulin. There are 5 classes of antibody listed here, where Ig stands for immunoglobulin, another name for antibody, followed by M, A, D, G, or E. These have different roles and locations. Some of these are monomers, some are dimers, some are even pentamers, depending on how many antibodies come together. But in any case, antibodies tag their specific antigen when they find it, so that it can be destroyed later. We will get more specific about this process when we look at particular infectious diseases. For now, let's continue on and switch over to the cellular immune response. Here we will look at T cells. These operate a bit differently, as activated T cells have the ability to kill cells of the body that have been infected by viruses or bacteria, as well as cancer cells. These cells are more diverse and complex than B cells, but they come in two major types, CD4 and CD8, which refer to glycoproteins that act as surface receptors, though they differ from antigen receptors. Rather, they interact with other cells. CD4 cells activate B cells, T cells, and macrophages, while CD8 cells destroy foreign cells or body cells with foreign agents. T cells undergo activation and differentiation when T cell antigen receptors interact with antigen-presenting cells. Then co-stimulation must occur from other molecules on the surface of the antigen-presenting cell. This leads to proliferation and differentiation. The resulting T cells can be of a wide variety.
Fate of antigen after penetration. Antigen is confronted by immune system in three routes. Antigen entering through the bloodstream is carried to spleen for interaction with antigen-presenting cells. T cells are activated by TCRs and B cells by BCRs and cytokines. Antigens may lodge in the epidermal, dermal, or subcutaneous tissues to stimulate inflammatory responses. They are transported through afferent lymphatics to regional draining lymph nodes where APCs interact with T and B cells to generate an immune response. The antigen may enter the gastrointestinal or respiratory tract where it lodges in the mold and interacts with macrophages and lymphocytes. Also, lymphocytes entering the efferent lymphatics are carried through the thoracic duct to the circulation and redistributed to various tissues.